I have your scantrons. They've all been scanned. All the scores have been uploaded to D2L. I will try and have those scantrons for you on Friday. So if you'd like to pick them up, you can certainly do that, or come find me after class. Another couple of quick announcements. The first one is our biology department seminar this week, molecular explanations for the toxicity of oxygen. So why oxygen is generally a really bad thing as far as most organisms on this planet are concerned, and how we and a few other organisms actually deal with it, which should be really, really interesting. The second one is a shameless plug for OMSI After Dark. It's Alive, which will be this evening, featuring viruses, dead or alive, with somebody you may know who will actually be there. So there's lots of other things which are there. Another one of the programs that I do is this Meet a Scientist, just in case you felt you hadn't met one already. This is a neat program that OMSI does as well. So, and if you come to this, at least you can drink beer and ask me about whether viruses are alive or not, and we can have an interesting discussion. I'm not supposed to, which is the only downside of this particular event. So, with that being said, let's continue a little bit with our last favorite bacteriophage, and then talk about our next favorite bacteriophage, and then we're done with bacteriophages for the term. Sniff, sniff, sniff. I have actually really seriously thought about doing a whole quarter on bacteriophage. I think it would be really cool and fun in my copious free time. So, that being said, let's get right into sort of finishing up with bacteriophage T7. And again, we talked about origin, we talked about structure, we talked about binding and entry, we talked about transcription and replication. All that stuff was on the exam, so we've forgotten it, right? But I did want to mention really briefly the biotechnology applications for bacteriophage T7, because this is what probably the vast majority of you, and certainly the vast majority of researchers today, know about bacteriophage T7. They use a particular enzyme that we've already talked about quite a bit already. And then, of course, we need to finish up talking about how genomes get out. And we already talked a little bit about the concatemers. We'll talk more about concatemers today as well. And just the whole idea of the packaging process. When you have a concatemer, it's literally multiple copies of the genome just hooked up head to tail all the way along. Again, many, many copies thereof. And we'll look more at this when we talk about bacteriophage Lambda later on today. And the idea is that you have one copy of the genome. That copy of the genome gets put right next to the head structure. That's the portal protein. And then one copy of the genome gets put inside the head. That gets chopped off. That piece of the genome, the end of these multiple copies of the genomes, finds another head to package into, etc. The head itself, just like we already saw in terms of Phi-X 174, and true for pretty much all of the bacteriophage, have scaffolds. And we haven't talked about all the proteins that are involved in the scaffolding. The take-home message here is that we have an internal scaffold that then has to be got rid of. How do you get outside of the cell? Again, these infect E. coli. So you've got two membranes that you need to get through. And there's a very hydrophobic protein which just makes holes in membranes. And you get enough of that, it makes a hole in the membrane. That's the hole in the internal membrane. Then there's the T7 lysozyme that we already talked about before in terms of how it interacts with the polymerase, which is an interesting story that we'll get back to in just a second. But it also has this enzymatic activity. Lysozyme just breaks up peptidoglycan. So you make a hole in the inner membrane. That allows the lysozyme to get into the periplasmic space where it chews up the peptidoglycan, and the cell falls apart, and you have lots of wonderful T7 that thing can go out and walk around on its tail fibers and try and find the right place to infect. So that brings me to this whole idea of why the vast majority of molecular biologists know about T7 and have no idea it has to do with a bacteriophage. It's all about that T7 RNA polymerase that we mentioned. And why? Okay, who are we at now? Gino? Is Gino here? Yeah. Why is the T7 RNA polymerase so wonderful? Yeah, the structure is similar to DNA polymerase, and what does that specifically mean in terms of the number of subunits that that polymerase has? Yeah, exactly. It has one subunit, and that makes it really useful for biotechnology applications because you just have one protein that you need to make, one protein that you need to put into a tube, and put a big price tag on it and say, hey, you can buy this RNA polymerase. And has those very specific promoters, which are different than the promoters that the cellular DNA-dependent RNA polymerase use. And so what that means is you can use this particular polymerase that also goes much faster than the cellular polymerase to express what I always like to call YFG, so your favorite gene. And so here's gene X right here, which we could also call your favorite gene that you put into E. coli in a very specific vector, so an extra plasmid. And that plasmid, they're called the PET vectors. Any of you worked in various different labs and tried to express recombinant proteins. Very often you use these PET vectors. And the key with these PET vectors is that they have a promoter for the T7 RNA polymerase. So this is a very specific promoter. It's only going to be expressed when you have T7 polymerase around in order to be able to make the messenger RNA, which eventually will get translated and make the rest of that gene product. So you have this vector, then you have the T7 RNA polymerase, usually expressed somewhere else in your host chromosome. Very often this will be a particular kind of E. coli, very often called BL21. You may or may not have heard about BL21s, but those are E. coli that have the T7 RNA polymerase in their genome. So all you need to do is put your favorite gene into one of these PET vectors, which has a T7 promoter, and then you have the T7 RNA polymerase. You express this, i.e. usually by putting in IPDG, often under the LAC promoter. That will make lots of this polymerase. That will then bind to the promoters and make large amounts of messenger RNA, which will get translated and make your favorite protein. So this is a really nice technique to make large amounts of your favorite protein. And I would guess something like 80 to 90% of people who are making recombinant proteins will use a vector like this because it works so well. There are a few issues with this, and that's particularly if you're trying to make protein in E. coli that the E. coli doesn't like. And to deal with that, there are a couple of different ways. One of those is to include this T7 lysozyme. So we just mentioned T7 lysozyme is important for breaking down the peptidoglycan, but it also can repress the activity of the T7 polymerase. So if you have a small amount of the T7 RNA polymerase around and a small amount of lysozyme, this is not going to bind to the promoter. It's not going to make the messenger RNA for your favorite gene. As soon as you start to make more of this, now you will start to make more and more of this. And so the idea here is that your background level is relatively low. So you have a gene that's toxic in E. coli. You can suppress its expression by having a little bit of this T7 lysozyme around. And then if and when usually you have the E. coli that's very happy, then you say, OK, just make a whole bunch of this protein that you don't like. And there you go, and you'll make a lot of this. The other thing which these are used for is for making large amounts of RNA, and just RNA in general. And this is particularly true if people are interested in studying that particular RNA. Very important for people who are doing things like RNA evolution, particularly Dr. Lehman in the chemistry department here at Portland State. So he has, instead of a protein encoding gene here as his favorite gene, he actually has a ribozyme here, which is a RNA which has enzymatic activity that is being controlled by one of these promoters, T7 promoter, for instance, which will then make a large amount of this RNA. And I think he goes through probably thousands of dollars worth of T7 polymerase in any given year to make all of this RNA. So it's a great way to make RNA as well. So again, most biotechnologists these days, when they think about T7, all they think about is T7 polymerase so that they can make lots of RNA. And they forget that it's part of this really fascinating virus structure as well in replication system. I mentioned the fascinating virus and virus structure and replication system, partly because we also know a lot about the structure of these particular viruses. This is actually not T7. It's a very closely related virus to T7. Also has these tail fibers that we saw associating with the various different receptors on the surface of the cell. But this one, I think, is much more fascinating from the point of view of looking at the DNA on the inside. So all of these black lines here represent the DNA packaged inside one of these virions. And what should hopefully be really obvious here is this is incredibly tightly packed into this structure. And it's pretty amazing that you can have all of this negatively charged DNA being so tightly packed into a structure. And as I believe I mentioned, but I may have forgotten to, is the packaging machinery through this portal protein is an ATPase. And so it expends a large amount of energy to package this DNA into this structure, which is otherwise energetically an inefficient process. And then, as soon as you have the release of this DNA, of course, 850 so nucleotides come out. That starts to get transcribed and pull the rest of it inside the cell. You have a question, Nicole?
Yes, so the core proteins, since they're in control of getting that first 800 and some odd base pairs out, do they also control the stability and the structure of the DNA being inside the capsid or inside the virion? So just because it looks so even and everything, the way that it's laid out in there, it's weird that it can do that.
Okay, I'm not sure I completely understand the question here, but I believe, again, correct me again if I'm wrong here, is that it's that structure, so it's the E version of these core proteins, you're exactly right, that then come out, make a hole through the membrane, and allow the genome to come out. It's probably the pressure which is generated by all the DNA being packaged in here that will keep it that way until basically you break out, and they call this structure here, and so this is, again, a very high-resolution structure. This is a zoom in right here of this junction point right here inside the portal. They have this valve, and the idea is that as soon as this interacts appropriately with the receptors, this valve is opened, and this basically gets squeezed out in that process. What you can look at is that if you now look at an empty particle after that genome has been released, you just see the portal protein. You don't see any of the core or the DNA. So all of that has come out the bottom here. Yeah? So where does the portal protein get its energy from? It's an ATPase, and so it just uses cellular ATP. So it's just ATP hydrolysis, ATP to ADP. So a classic kind of ATPase. And it turns out that these proteins, the portal proteins, and we'll talk a little bit later about the terminase proteins, these are some of the most active ATPases in any cell. They're really amazing molecular motors. Because if you think about packaging this amount of DNA inside this structure is really pretty amazing that that even works at all. And so it definitely requires large amounts of ATP hydrolysis. And I forget how much it is. I think it's like 2 ATPs per base pair or something like that. It's massive. It's, yeah, huge amounts of energy expenditure. Yeah, Mario. Pardon, I missed the end of that. Oh, are there any extra proteins that are involved in packaging the DNA? Not in the case of these bacteriophage, but we're getting ahead of ourselves about a week when we talk about some of the other viruses. It's the same kind of problem. You have to package your DNA somehow. You have to package that nucleic acid somehow into the relatively small capsid. And how you do that, it's evolution. You just have to have something that works. In this case, it's those ATPases that are going to burn huge amounts of ATP and just stuff everything in. In some other cases, it's actually a very different kind of packaging process which takes place. And we'll talk about those when we get to the other viruses later on. But again, the whole compaction process, getting that amount of DNA inside one of these particles is hard. And there are lots of different ways of doing it. And this particular one, again, is ATPase, just by pumping things on the inside. Oh, yeah, sorry. James.
During the packaging process, does
So the question here is, again, correct me if I'm wrong, is basically how is this packaging taking place? And it takes place literally by starting at one end, and that's where the end of your genome is. And that's recognized by some other proteins that we haven't talked about here. It'll be the terminase in the case of Lambda. That then will bind to the procapsid, which is not shown on here, and then from there it will package everything in until it gets to the end of the genome. That will then get cut. The next genome will come into the next prohead, and it will get cut and packaged again. So it is a very specific in this case. There are some viruses for it, which is a little less specific, but we're not actually going to talk about those because I'm not doing a whole term on bacteriophage. Again, I wish I could. So just, again, overview. We talked a little bit about where T7 came from. It was that commercial preparation, which was Alex Paulo. No, not here. Kietron. Question on the exam that everyone got right, Nicole?
What was the question?
Where did bacteriophage T7 originally come from, we think? The commercial preparation.
That was from phage therapy.
Was from phage therapy, exactly. Then the structure. Oh, this actually I forgot to mention here. These are actually nice icosahedral capsids to look at the T equals 7, because you need to go from one 5-fold axis to the next 5-fold axis. It's actually easier to here to see on the empty capsid. 5-fold axis to 5-fold axis. You go 1, 1, 2, and that gives you the T equals 7. Although I'm sure all of you just memorized it because it's T7 is a T equals 7 icosahedron. So the phage T7, again, how's it get in? We had the little tail fibers walking across the cell. Transcription, we've hopefully done to death the T7 RNA polymerase now, but also that's how it's getting pulled inside the cell, is through that transcription process. And the replication, again, it uses the T7 RNA polymerase as a primer, and then has otherwise almost all of the its own replication machinery, the T7 DNA polymerase, T7 primase, et cetera, and we just talked about the biotechnology applications. So any more questions on T7 before we talk about, again, bacteriophage that we could spend the whole term talking about? So bacteriophage Lambda. If you're interested in more information on bacteriophage Lambda, I have this small book up here. Lambda 2, it's actually the second book written on Lambda. All of these are various different experimentation which has been done on the Lambda bacteriophage. And if we get through all the rest of the slides today, we'll take a look at a nice DVD that was put together in terms of looking at this, say, pinata of paradigms. I love the title for this chapter. I think it's really fun. But again, it's just we're getting larger and larger, and also thinking more and more about different kinds of replication patterns. So bacteriophage Lambda is a double-stranded DNA bacteriophage. Again, should sound very familiar. Paradigm, lots of different kinds of processes. A lot of what we know about molecular biology comes from the study of bacteriophage Lambda. Huge amount of that is looking at transcriptional regulation. So transcriptional regulation in bacteria, how transcription is being regulated. Much of what we understand in terms of thinking about transcriptional regulation comes from the study of Lambda. It was one of the very first to be studied. And the big difference here, a nice little red part, again, special thing about this particular kind of phage, is this whole process of lysogeny, whereby the viral genome gets into the host genome and hangs out. And probably the majority of viruses in the environment, again, all those little dots that we saw way back when in lecture 1, that is going to be derived in many cases from these lysogenic viruses. Now, you wouldn't see those as little dots. They're actually going to be in the bigger dots because that's where those genomes are found most of the time. To get lysogeny, in most cases, it's actually going to be through a genome integration process. It's not always integrated. And this is just the cartoon. Kind of a shout out here for Viral Zone. Did anybody look at Viral Zone? Actually quite nice for the individual virus families and looking at those. These are actually images from Viral Zone. And here you can see very clearly, hopefully, that this is also a T equals 7 icosahedral capsid structure. A couple key concepts as far as excuse me, Lambda is concerned. Anti-termination. What the heck is anti-termination? Has to do with transcription. A lot of the transcriptional regulation in Lambda has to do with use or non-use of transcriptional termination sites. Feedback loops, this should sound very familiar based on that other course last term. Very much having to do with positive feedback loops. Once you make a decision, and we'll talk about the genetic switch a little bit later on, then that decision is maintained. And so some sort of cellular memory, or in this case you could say a viral memory. And again, this lysogeny idea, this is that a virus, which will be called a temperate phage in this case, can either lyse cells immediately, or it can wait and potentially lyse cells later on. And again, a lot of this has to do with the integration process. So our standard look here at Lambda, where'd it come from? So structure, binding and entry. Mostly going to talk about transcription, lytic growth, i.e. how the lysis pathway happens, how lysogenic growth happens, which is hanging out inside the cell, and then the choice, whether to grow lytically or grow lysogenically. And then we'll talk a little bit about replication and the release, although a lot of these things are very similar to what we've talked about before. This is an electron micrograph of a Lambda bacteriophage. So it's got this icosahedral head and a long flexible tail. And these flexible tails are pretty unique to this whole class of viruses. And it makes nice plaques. This is an example of Lambda bacteriophage plaques here. Turns out this is Lambda bacteriophage plaques from a particular mutant strain of Lambda bacteriophage that we'll get back to a little bit later on. So where did Lambda come from? Lambda was basically a mistake in terms of finding it. Nobody knew that the standard E. coli that everyone had been using literally for decades, E. coli K12, had a virus hidden in it. So this was a lysogen. Actually had the virus genome hanging out inside the host genome, and then only under very specific conditions, some plaques were formed, and this is basically was a control. Somebody was looking at infecting their E. coli with a different bacteriophage. This, I think, was Esther Lederberg, who found this. And that was that you were trying to do a negative control for their other bacteriophage infection, and then they had these cells that they just put on the lawn, they're uninfected, and then lo and behold, they started to see plaques. It's like, wait a minute, you're not supposed to see plaques here. And eventually what they found out was that there were some strains of E. coli, in fact, the standard strain that people were using, that could make plaques on a slightly different kind of E. coli. And it turns out the E. coli was actually a mutant, which had lost the copy of the bacteriophage in its genome. And when you look at lytic growth, actually looks pretty normal, standard one-step growth curves. And this is this whole process right here. And I will be looking at this particular cartoon many times throughout the rest of this lecture. You have an infection, circular genome. That circular genome here, once it gets inside the cell, again, this is E. coli, can either undergo lytic growth, where you make a lot of DNA, you make a lot of the proteins, you have lysis, or undergo this lysogeny process, whereby the viral genome gets integrated into the host genome and then persists over cell division. And this literally can go for years and years and years. And then at some point, you have this induction step that takes place. And this induction then leads to lysis. The key here is that these cells are actually now resistant to another infection. So if you just try and infect one of these lysogens, you're never going to see virus production. And so that was the key, again, with Esther Lederberg, is that she was looking at a mutant which was lacking this copy of the genome. We'll see why that is a little bit later on. The fact that this particular virus can replicate in either a lysogenic process or a lytic process means it's a temperate phage. It's not a lysogenic phage. 90+ 90+% of a lot of the population, even in a grant proposal, which I just reviewed, calls these lysogenic phage. They're not lysogenic phage. They're temperate phage, which means they can be lysogenic or lytic. It's the cell itself, which is a lysogen. And if you just think about the word, what's lysogen mean? It can generate lysis. But that's the cell that generates lysis. It's not the virus itself which is generating lysis. Does that kind of sort of make sense? Okay, so the this is you have a lysogen, which would be the bacteria, which has this potentially lytic virus in it. Anything that is not a lysogen is not going to produce lysis. There's no name particularly for a cell that doesn't have a lytic phage in it. Yeah?
So is the integrated machinery coming from the cell itself, or is the bacteria phage bringing them with it?
Great question. We'll get to that a little bit later on. So the integration process is all virus-mediated through a interesting protein called integrase, again, ridiculously creatively named. So what does this genome actually look like? And actually, well, I'm getting a little out of order here. But now that you mention, how does it get in? It's a site-specific recombination process through the Lambda integrase. And all of that was shown by Alan Campbell here, who I had the great pleasure to hear give a talk about 2 years ago, actually 3 years ago now, at the 100th anniversary of bacteriophage. Unfortunately passed away about a month ago. I also had the slightly less amount of pleasure to have him review one of our papers, which he completely trashed, which was for very good reason, actually. But yeah, really very bright, helpful, but direct bacteriophage researcher. So he was the one who discovered that this integration actually took place. And through the whole process of site-specific recombination, which of course is now in all of our textbooks. Another person who you may or may not have heard of, Hershey, as in Hershey Chase, who was looking at bacteriophage as being indication that DNA was the genetic material. He also worked on bacteriophage Lambda, and what he found is that even though this genome was linear when it was inside, and linear just having one end and the other end when it's packaged inside the virion, as soon as it comes inside the cell, it circularizes. And the way that it circularizes, also shown by Hershey, is that it has these so-called cohesive ends, or COS sites. And all that means is it's overlapping with the two different strands, different overlaps. In this case, it's 12 nucleotides that can now base pair to each other. When it comes in, the very end of the genome has this single-stranded piece, which is complementary to a single-stranded piece at the other end of the genome. So as soon as that whole genome comes in, these guys get linked up to each other. 48,502 base pairs. Why do I emphasize this? No, it's not going to be on an exam. But it was, I think, the second complete genome sequence to be determined. And so now we can go down our list again. Bridie, Cawthorn? Yeah. What was the first genome sequence to be done? There's also another phage. Yes, very good. We haven't talked about that many yet, so we're getting close. Exactly, PHAX-174. Also by Fred Sanger, who ended up getting 2 Nobel Prizes, one for protein sequencing and one for DNA sequencing. And so they did PHAX-174 and they did Lambda, and that these were the first ones they were doing. Also gives you an indication on how important they were for understanding a lot of molecular biology at the time. After they'd done those sequencing, and actually around the same time, because they were doing a lot of genetics as well, what they found is that this is not, again, unlike the case that you have with bacteriophage T7, that the genome is set up in these really interesting block-like structures where you have the structural proteins, head proteins, tail fiber proteins that we'll talk about later, all of these that are linked together. You have all of the proteins that are involved in recombination, all linked together. These regulation proteins, and these are the one we're going to talk most about here, and then finally some lysis proteins. So every sort of set of genes that you need for any given part of the virus life cycle is all put together in these nice little, excuse me, they like to call them gene block structures. So let's look a little bit first at the structure before we talk about how it's made. Again, these have a T equals 7 icosahedral head structure with a portal protein. This tail is actually a little bit different. We've talked about helical tails before. This is actually not a helical tail. It's a little bit different. It's a whole bunch of donuts, basically, that are stacked on top of each other. And that process is probably a lot more flexible in terms of the tail and then the tail interacting with various host cells. This is actually a slight change from what you see in your textbook. In your textbook, it doesn't have these extra tail fibers that are hanging off at the end of bacteriophage lambda. Turns out that for many, many years, the researchers were actually working with a bacteriophage lambda that was a mutant. It was actually lacking the tail fibers. And so it was only literally in the last probably about 20 years that people found that lambda really did have tail fibers. And so it looked a lot more like some of the other bacteriophage, just the mutant, which still worked, had this flexible tail, and it just had lost these particular tail fibers here. This is an example of the bacteriophage head. It's actually quite hard to tell that this is T equals 7. If you want to challenge, go ahead and take a look at it. The receptor for lambda was actually determined way before we knew what the real use of that particular receptor was. We know it's a maltose transporter, but it was originally discovered as Lambda. Why called Lambda? Because it was all the geneticists, and what did the geneticists do? They break stuff and try and figure out what they broke, and in this case, it was lambda binding. So if you had a mutation in this gene in E. coli, it couldn't bind to lambda, and so that was how it was originally described. One other thing which is quite important about this, if you ever try and grow lambda bacteriophage, you have to make sure you're growing your E. coli. Your E. coli, you have to give it maltose. Because if you don't give the E. coli maltose, it doesn't make this maltose transporter protein. And if it doesn't make the maltose transporter protein, the virus can't bind to the cell, and so it can't infect. So it does turn out to need this particular protein, and again, that protein is being made normally. So this is a structure. Binds to the cell through this lambda binding protein, get a maltose transporter. Maybe it's that transporter that helps the genome come in. That's not entirely clear. And then once that genome comes in, it gets circularized. And at this point, once that genome's come in, it's been circularized, then the decision has to be made. Are we going to lyse our poor cell that we've come into and make a whole bunch more virions to go out and infect more cells? Or are we going to insert our genome into the host and hang out as a lysogen? Again, that's the cell with the virus genome in it. So the easiest thing to think about is to think about lytic growth. This is sort of the default state that happens with bacteriophage lambda. So again, I'll have our little cartoon up here in the corner. We're just talking about lytic growth here. So again, the important bits as far as we're concerned for regulation are all right here, this part of the genome, which we've just zoomed into right here. In this part of the genome, there are two major promoters, and again, incredibly creatively named PL and PR for the left promoter and the right promoter. Soon as this comes inside, the virus genome is circularized inside the cell. You have transcription from PL in this direction, and you have transcription from PR in this direction. Gets to these terminators, terminator R1 and terminator L1. So relatively short transcripts which are made. The most important of these proteins that are made by these transcripts is really this one right here, the N protein. Now, why the heck would you call something N? Why do you have all these letters in terms of a genome and have nothing to do with how the structure or function of any of these things are? I'll let Dylan Willis make a guess. Dylan's here. Amadreza? No. Kate Cochran, any guesses? Yeah. And this is a guess. Why would you just letter something? Kind of like maybe like the numbers we had in T7. Where did all those things come from? Order they were discovered, and how would you discover something like this, particularly if you didn't have a DNA sequence? Yeah, so genetics. So these are all genetics processes. So it's literally doing various genetics and how these genes are related to each other and just what the orders are. And literally by doing lots and lots of genetic crosses. You have one mutant, you have another mutant, how does that one relate to itself? And so a lot of these, again, T7, all the numbers, all of the bacteriophage lambda, various different letters, with a few exceptions, but again, for the most part, just letters and how they were found in terms of the various different mutants. And unfortunately, it's really kind of a mess, and you just have to remember them. Again, I didn't make this up. Trust me, there's a whole book written on it. So yeah, so the N protein. It's the N protein which is the most important in terms of lysis at this very first step. What does N do? It's an anti-terminator. What the heck is an anti-terminator? So an anti-terminator is a protein. Again, this is this N protein which gets made. It interacts with the cellular now RNA polymerase in order to say, hey, ignore this termination site. And in order to ignore this termination site, you also need this N utilization signal. Okay, the NUT site. So N utilization is a sequence in the RNA which basically tells the polymerase, again, to totally over-anthropomorphize here, slow down a second, and if the N protein is around, it can associate with the polymerase and say, okay, you're now a super polymerase, and you can ignore any kind of terminators that come up after this. And so that's what happens. You have anti-termination, which now allows this transcript to be made a lot longer for PL. Now, PR is more important because instead of terminating at this point, it will transcribe all the way through this Q protein. Now, Q protein works in an extremely similar way to the N protein as an anti-terminator. That anti-terminator now, instead of working on the PL and PR promoters, works on what people will call the major late promoter here, PR prime, which then allows us to transcribe all of these different genes. And if you go back up here, this is where the Q protein is, Q gene. You can transcribe, and remember this is circular, so it will transcribe off of this end and transcribe all of these genes. What are all of these genes? These are the head, tail structural proteins. Again, the late proteins which are important for making the virion. So this clearly is going to lead to making of all of these late proteins. Okay, happy with lytic replication? Are we happy enough to answer a clicker question on them? Good. So we'll do the same thing as usual here. And start. Oops, where's my, oh, no base station. It doesn't think my base exists. Okay, let's start this process again. Here we go. iClicker. It's already running. No, it's not running because you're giving me a hard time. Here we are. Okay, finally. I know you've all been missing out here. Okay, so our clicker question. So again, you have a minute to answer by yourself, and then we'll have some time to talk about it, and then we can move on from there. Or if everybody gets it right the first time, then we don't have to have a discussion. Stetson's lazy when he comes up with answers for questions. Let's see, there were 59 people here on Monday. I know that because I looked at all your exams. Okay, we've got a close to consensus here. Let's see if we can get a 100%. So everybody, talk to your neighbors, tell them what you decided and why. Ready to go again? Yes? Everyone ready to go again? Believe it or not, we actually had at least one answer for every single one of the options the first round. So, again, feel free to talk to your neighbors here. Make sure that your clicker is working the way that you want it to. Vote early, vote often, which does remind me, if people haven't registered to vote in Oregon, they should definitely do that. There's a primary coming up in May. So register, 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 vote, vote, vote. Not just your clickers. It'd be so much easier if we just use these for elections. I think we've only got 41 people here today, so the discussion does seem to make a difference, so believe it or not. Now, and also seems to make a difference in the correct direction. So it's about anti-termination is really about where you have this. So it's the N protein and the Q protein, which are both anti-terminators. So it's all about that process. It's not transcriptional elongation. There are some things which regulate transcriptional elongation per se, but that has much more to do with elongation rates rather than the actual termination. Yeah, Jacob. What does the anti-terminator look like? The anti-terminator protein or the process when it's actually going through the anti-termination? Yeah, so send me an email and I can send you a link to the crystal structure. So it has to do, so the N protein is associated with the RNA polymerase, and it changes the process of the termination. Now, what I don't remember whether this is a row-dependent or row-independent terminators that we're talking about, and that I would have to go and look up to see. But yeah, the whole, this is known to almost atomic detail exactly how it works. So yes, it's about transcriptional termination. Okay, so we talked about lytic growth. Let's talk about the other way that these things go. And if we can get the slides to move, that would be nice. We're not moving. Oh, how nice. Sometimes technology is just so wonderful, isn't it? There we go. Okay, so we talked about lytic growth. Now let's talk about what happens in lysogeny. Why, again, to totally over-anthropomorphize, does the virus want to, instead of trying to make a whole bunch more virions, hang out? And the main reason seems to be, well, if the cell doesn't have enough energy, metabolites, etc., then there may not be enough to make a whole bunch of virions, but potentially even more importantly, there may not be any other cells to go and infect. So and again, we'll talk about how this happens, the unhappy process. There's a protein called the Lambda repressor, also called C1. Now, I want to put somebody on the spot again here. Let's see. We had Samuel Nelson? Yeah. Yeah, so why is it called C1? Why is the repressor called C1? No idea. So we talked about the lettering before. So lettering usually comes from what? Genetics, right. And so I've mentioned a few times, what did geneticists do? Break stuff and try and figure out what they broke. And they often will name their mutation after the phenotype. So what kind of phenotype might you have here? What might C stand for? So I mentioned those plaques right at the beginning that are actually from a mutant form of Lambda. What might C stand for? Guesses? Well, actually, C2 and C3 we'll get to a little bit later on, which are named for exactly the same reason. Clear. So they're clear plaques. Now, that's a challenging question. But why are they clear plaques? Why clear plaques? Any idea? Yeah. So if you think about, just think about this process. So here, lysogeny is the cells are going to grow after you have that infection. So if you have lysogeny, what is going to happen? Are the cells going to grow? They've got this virus genome in them. As I mentioned before, that actually protects against future infections. So if you have a normal Lambda, you have what are called turbid plaques. And what that means is the cells that are in a plaque that otherwise would be killed, are those going to be happy cells? Heck no, they're going to be really unhappy cells because there's all these virions around. And so what will happen is some of those cells will now go through this process and be lysogens and be resistant to infection. And so you have a little bit of growth. And so these are what are called turbid plaques. And so if you have a C1 mutant, what happens? Are you going to have any of that lysogenic growth? No. So everything's going to lyse. So any one of the C proteins, C1, C2, or C3, those are always going to give you lytic growth when you get rid of those. Okay, so C1 repressor. What does C1 repressor do? It actually, one of the main things it does is protects against future infection. If you've got C1 around, you're not going to have any kind of extra lytic growth that happens. So what's known now is very clearly how the C1 protein works. It's a DNA binding protein. In fact, one of the very first, I think it'll be the second DNA binding protein to ever have been characterized. And it binds to these sequences around, and I should have had the genome up here, PR. And PR should sound really familiar because of PR and PL. Those are the ones which are leading to lytic growth. Okay, well, if you have C1 around, it's going to bind to these binding sites which overlap PR. Well, if you've got a protein overlapping PR, are you going to be able to have transcription from that promoter? No, because you're going to be blocking the RNA polymerase from coming down and binding at that position. So the presence of the Lambda repressor will bind. Turns out there are 3 binding sites for Lambda repressor, and again, creatively named repressor binding site 1, 2, and 3. These are the operators. Operators goes back to the operon model that Jacob Monod came up with for transcriptional regulation. So operon is what's going to be controlled by binding of some kind of protein to a particular site. And these operators are the DNA binding sites. Binding to OR1 and OR2 blocks transcription from PR. But one of the other fun facts about C1 repressor, and why is it in quotes, because it's also an activator of transcription. But it activates its own promoter. And this is the PRM. PRM, what the heck does RM stand for? Repressor maintenance. So Once you have lambda repressor around, that will stimulate transcription of the lambda repressor gene, and that leads to, who are we next? Leanne Harada? Leanne here? Yeah. Yeah, so if you have the lambda repressor, again, quote unquote repressor, activating its own gene, what process does that generate? What's it called? Pardon? So it's expression, but it's expressing its own gene. And so what would that be? It's a feedback loop. What kind of feedback loop? Positive, because it's expressing its own gene. So exactly, positive feedback loop. Exactly right. So the idea here is you have this positive feedback loop, which is going to lead to more and more production of the lambda repressor. Turns out after you get to a certain point, that's kind of counterproductive. And so then there's actually even more repression that happens, which blocks the PRM promoter as well. And the way that this works is just that the binding for OR1 is much better than for OR2, which is about the same as OR3. So you have this positive feedback loop until you have enough of this lambda repressor to block absolutely everything. So this is great, this is wonderful, but once you've got all this lambda repressor around, how the heck can you actually do lytic replication? At this point, it seems like you're pretty well shut down. You have this positive feedback loop. You know, what's happening here? So what happens in terms of induction, which is here, you've gone from this lysogenic state, and we will talk about integration. Don't worry, we'll get there. How do you go now to a lytic state? And what happens here is you just get rid of C1. And getting rid of C1 is a proteolytic event. And again, proteolysis is going to come up again and again and again in this course. And this is when the going gets really, really bad. And so how does a cell know that the going is really, really bad? Usually this happens in terms of DNA damage. So it's a DNA damage response. And let's see, well, we'll do this, continue to put people on the spot here. Daniel, why would DNA damage be a real problem if you're in a lysogen? Lose what kind of information? You have DNA damage. So DNA damage is going to happen as you're a lysogen. Why would you want to not be a lysogen with DNA damage if you're a virus? Again, totally other anthropomorphizing here, but why? Why would DNA damage be something that the virus does not want to deal with? Why would it want to go lytic at that point? Yeah, well, so if the DNA of the virus now gets damaged, it's clearly a real problem for the virus. If the DNA in the rest of the genome gets damaged, it's a relatively small genome. It's 50,000 base pairs. The E. coli genome is about 5 million. So if the DNA damage response is already happening, hopefully it hasn't happened, again, thinking this totally from Lambda's point of view, it hasn't happened in the Lambda genome yet. So if there's DNA damage happening, there's probably going to be more DNA damage happening. So it's time for us to get the heck out of here. So that seems to be the reasoning behind why you have this induction after DNA damage. So DNA damage happens somewhere in the cell. That DNA damage is sensed by the cell. The lysogen senses, hey, there's DNA damage happening. I'm a DNA in this genome. This is not a good place to be. It's time to get out of here. And so that process is what's called the SOS response, so Save Our Souls response. And again, it's a response to DNA damage. And if there's a DNA damage that happens, usually Rec-A, pardon me, let's go back. Rec-A is the single-stranded DNA binding protein, really important for homologous recombination, repairing DNA damage. That seems to be the homologue of this repressor, which represses these genes, because you don't want to turn all these DNA damage genes on under normal conditions. So that's repressed by Lex-A. That Lex-A usually gets cleaved by Rec-A when there is DNA damage. Lex-A, it turns out, has a very similar structure to Lambda C1. So Rec-A will cleave Lambda C1. You cleave Lambda C1, can't associate with the DNA anymore. So you turn on PR and PL, make N, make Q, and make lysis happening. Yeah?
So is this, I guess, kind of two questions. Is the SOS response triggered by specific DNA damage in the host genome? And then how does the virus genome know that DNA damage is occurring?
Okay, so the SOS response is actually a very general response. So to DNA damage, and it turns out other kinds of damage as well. And all of that's regulated by LEX-A. LEX-A is a bacterial repressor. So it binds to all the promoters for all of these genes that are important for DNA damage repair. And then as soon as that is degraded through Rec-A, which is sensing single-stranded DNA for the most part, then you'll end up turning on all of these other genes, which also includes cleavage of LEX-A. And that cleavage of LEX-A, since the structure of C1 is similar to the structure of LEX-A, that proteolytic cleavage will happen to LEX-A and to Lambda C1. Yeah, do you have a question? Yeah, so I guess paraphrase your question, Joe, but I think that your question is, so it's going bad when they first come in, then they go lysogenic, but it's only if it's going really bad that they want to get back out. What's that switch over? Yeah, and so basically the first step, it's much more of a metabolic case. And again, we'll talk about this in just a second right now. It's metabolism. So metabolism is going relatively slowly. The cells are not growing very quickly. But they're not undergoing active DNA damage. And again, if you think about what David was mentioning, that it's the DNA damage, you don't want to damage your own genome. That's where it's really clear that the virus does not want to be there anymore because DNA damage is happening. So DNA damage is clearly something where the virus says, this is bad news. It's time to go and find a cell that's not undergoing DNA damage. Yeah, Margot. So how does it find, basically, I'll paraphrase your question, is basically how does this SOS response work? And it mostly seems to be you have single-stranded DNA which is formed. As soon as you have single-stranded DNA which is formed, then Rec-A will recognize that and become active. And Rec-A is around all the time. But it's not activated until you have this single-stranded DNA. So Rec-A, there's a lot of Rec-A around, so it will cleave LEX-A, but it will also cleave C1. So this is happening at the same time. So the same time the SOS response is being turned on by cleavage of LEX-A, you're also turning on lytic growth or induction of Lambda. And it's just enough copies of the activated now Rec-A protein, but the key is activation of Rec-A. Yeah, James? Is there also induction when the cells are growing very well? If you do massive DNA damage, and the standard way of doing DNA damage here is UV irradiation, you can have happily growing cells, and you zap them with UV, the virus will be induced. So it's an independent process of the metabolic process, which we're just about at, which eventually we'll get to, right? I promised. So again, we mentioned how you grow lytically. That's PR, PL, N, and Q. How do you grow in terms of a lysogen? So all about C1. But how's this decision made? How do you decide that the cells are happy or unhappy, as the case may be? All has to do with the relative amounts of lambda C1. So it has to do with the concentration of C1, because again, C1 will block OR1. And this other protein called CRO. And I won't ask you what CRO is, because it's the control of repressor and other things. Again, horrible naming process here. But CRO is really another DNA binding protein, also a repressor of transcription, and it binds really well to OR3, which will block PRM, and poorly to OR1 and OR2. So in the presence of CRO, you have no expression of C1. In the presence of C1, you're not going to be making any CRO. So it's really about C1 versus CRO. And eventually we'll get this to move from one slide to the next. There we go. So it's all about the amount of C1 versus CRO. How do you get C1 and how do you have CRO? So mention that CRO blocks PRM. PRM is repressor maintenance. But how do you get repressor in the first place? This is a Lambda protein. So there's this other promoter, repressor establishment, which is, where are we here? Repressor establishment, which is positively regulated by this protein C2. C2 because clear plaques if you get rid of it. So if you don't have C2, you're never going to get C1. Again, clear plaques, you're always going to be going lytically. C2 is an activator. It will activate this repressor establishment, make C1, also makes the integrase gene, which at this point we're probably going to be talking about on Friday, and has this other really fascinating promoter here called PAQ. AQ. Well, we had PQ already. That's for all those late genes. PAQ is P anti-Q. So what anti-Q is, it's literally a transcript which is in the opposite orientation, and then we'll base pair to any transcript which is made by Q. Double-stranded RNA is then going to block any further transcription. And it turns out double-stranded RNA usually gets degraded pretty rapidly. So this antisense RNA is also part of the process. So then again, okay, well, it's all about C2 now as opposed to C1. Well, what about C2? How do you regulate C2? Now it turns out that C2 in and of itself is pretty unstable. And this is getting back to Joe's question about how does the cell sense if it's going good or going bad, metabolically speaking. So there's this C3 protein. Again, mutated, everything goes lytic. This will stabilize C2, but it gets broken down by proteases. And these cellular proteases, if the going is good, then you're going to break down anything that's coming in because proteases are going to be breaking down proteins, cells are happy. If there's not very much in these proteases, they're not going to be able to be active, and this is all metabolically speaking, then C2 is not going to be stabilized. You're never going to make C1. CRO comes in, and you go down the lytic pathway. Okay, let's see. We'll go and look at this very last step, and then we'll talk about integration on Friday. So C2 leads to production of C1, also leads to the production of the integrase gene. And integrase is, as Alan Campbell showed, a site-specific recombinase that will bind to the attachment site in the phage, the attachment site in the bacteria, and pop it into the genome. And then once you have the genomic integration that takes place, this is, of course, now producing C1. Any genome which comes in is now going to be exposed immediately to C1. As soon as you have C1 around, that's going to block any kind of superinfection. And so this is why no one discovered bacteriophage lambda for up to 30 years. So we'll talk more about integration next time and also try and show the DVD.